This video is about enzymes, and enzymes are just a specialized type of protein that does a specific task. And if you remember back from our protein chapter, we found out that there's a primary structure of proteins, which are those amino acids stuck together. The secondary is where they become the alpha or the beta pleated sheets, which are kind of like the zigzag pieces. Tertiary is just when that one protein is finished, so you can see the coils of the alpha helix and the zigzags of the beta pleated sheets. And then finally, you take a couple of these things and put them together in the right order, you're going to end up with your quaternary structure. And that's just going to be one protein that was made at one time, here's another one that made at another time, another one, and another one, and they all just kind of fit together, and now they're ready to do whatever job they need to. So this is how proteins come together, and it's exactly how enzymes come together. But what do enzymes really do? Enzymes are this thing that are called catalysts. And what a catalyst does is it just lowers the activation energy that's required to do some work. So this would be energy, and this is time, and basically you could see that the energy is reduced, and the time would actually be much, much faster with the enzyme. So what does a catalyst do? A catalyst basically speeds up a reaction to make it so less energy is required by the cell. Let me ask you a question. If the enzyme was gone or broken, would the work still continue? Well, that's kind of represented by this black line. This black line is without the reaction, without the enzyme or catalyst. So you can see that it still happens, it just takes a lot longer and requires more energy to do it. So, what is the enzyme doing? Basically, an enzyme basically has this opening that's very special, and it's called an active site. And that active site only allows one thing to come in, called the substrate. Now the substrate has a special opening, and the active site is a special opening, and these are the two things that can only fit in. Nothing else could fit in. When they do come together, a reaction occurs where the substrate is broken and becomes a product. Now, this could happen in reverse as well, where two substrates come in, and basically the enzyme puts them together, and that could be the product. But you can see with this one, we're going to go ahead and break something down. And you guys remember that to break something down is lysis, and how we're doing this one is we're going to put a water here, hydrolysis. So basically, this enzyme took the substrate and did hydrolysis, and basically made it a product. Would this happen if the enzyme wasn't there? Yes, it still would. The substrate itself would eventually break down into the two products, but remember, it would take a lot longer to do it and require a lot more energy by the cell. The enzyme is just the catalyst. And again, to specify, if this was the enzyme and this is the substrate, they only could fit together. That's it. So like for instance, if this was the enzyme called lactase, ASE, anything that ends in an ASE is an enzyme. And if this was the sugar lactose, OSE, these two can only fit together. If this was the sugar sucrose, then sucrose could not fit into the lactase enzyme, and we wouldn't get the reaction. So it has to be the pair comes together, and they have to fit together perfectly. So just, again, we have our enzyme, we have our specific opening called the active site. The thing that's going to come inside of it is going to be the substrate, and eventually when they fit together, it's going to be the substrate complex, but a product is going to be made. Enzymes are reusable, so you can keep using them over and over and over again. If you remember our lab where we took our liver sample and we added the hydrogen peroxide, the liver had this enzyme called catalase, and the hydrogen peroxide was able to, the hydrogen peroxide when it came to catalase was broken into two products. So that was going to be the water and the O2 gas. And you did it a couple of minutes later where you hit it with another 2 mils of hydrogen peroxide and it kept working. And you hit it with another 2 mils and it kept working. We could have waited until the end of the class and still done it one more time. It'd still be working because the enzyme itself will constantly be working. The only thing that stops it is no more substrates. So enzymes are like tiny little manufacturing machines. The only time they can make something is if they're a substrate, and they go ahead and make these products. And we know that anything that ends with an ASC is more likely going to be a enzyme. There are a few exceptions to the rule, but we'll get to those when you get to Bio 202. But sucrase is going to be looking for sucrose, and when they come together, they're going to make a product. Lactase is an enzyme that looks for lactose. Maltase is going to look for the sugar maltose, but it doesn't have to just look for sugars. They could look for fats or carbs or proteins, specific ones, or metals. So they have different jobs, but I just chose 3 sugars because I figured you know what sucrose and lactose and maltose was. Enzymes definitely help you do work faster. They help support life. Without them, your digestive system would take 10 times longer to work than it does now. And when we write what's going on with an enzyme, we kind of use this reaction where we have reactant plus reactant, and an enzyme will equal a product. So this is going to be one substrate plus another substrate come together in this enzyme to be a product. Would that be an example of hydrolysis or dehydration? So it looks like it's adding these two things together to become the product. So the only way you can do that is to remove a water. And would that be hydrolysis or dehydration? So to remove a water, it would be called dehydration. So this enzyme is doing dehydration. This one has a substrate or a reactant. It goes into the enzyme and breaks it in half. And to break something in half is called lysis. So, and you do that with water, hydrolysis. There are ways of stopping an enzyme from working, and we know that anything that denatures a protein is going to denature an enzyme because an enzyme is just a specialized type of protein. So the things that can cause it to stop working are temperature and pH, and there are a few other things that can stop enzymes from working too. We know that with temperature, like temperature and pH, when you move them outside of a certain comfort zone, what happens is that they're going to start to denature. If it's a little outside the comfort zone, it's going to be okay, but too much, it's going to cause a problem. That's why when you have a really high fever, it's dangerous, because your body really starts to cook proteins, and the one that we're worried about is your brain. That's why if you ever hear people that have a really high fever, they're going to have a, they could get brain damage. A cofactor, let me go ahead and show you what a cofactor is. A cofactor is something that is needed by the enzyme to actually do some work. It usually means, cofactor usually means something inorganic. So when you need those essential metals, what you really need is something to make an enzyme work. So if you're running low on copper or iron or magnesium, really it's that thing that sort of wraps around the enzyme, and that's the thing that's going to make it work. And a coenzyme is a type of cofactor, but it's usually organic. So this is going to be something that has a carbon in it that makes it work. So cofactor is going to be inorganic. This one's going to be organic. A type of coenzyme is going to be B12. Without it, vitamin B12, you guys now know, it's called NADH. It basically goes ahead and gives us energy. And the last one is inhibitors. And I know you can go online and look up what inhibitors are, but what I basically want you to know is There are active sites for enzymes, and there are other things competing for that active site. And if this thing called an inhibitor, which has the same opening or shape as that active site gets in, it'll actually cause it to stop working. So if the enzyme has the substrate come in first, it's going to go ahead and work. And let me show you a picture of that. Oops. Guess I got rid of the picture. But basically what happens is we have the active site, and the substrate's supposed to fit in. But imagine if there was something other than the substrate that had the same opening that could fit in and block this site. And if it blocked it, basically all these things that would normally fit in can't fit in. So one of the things that I think about when I think about these things that can block it is mercury. You guys know that mercury is a poison and you're not supposed to touch it or ingest it or do anything with it. Basically what happens is it has the ability to sort of morph shape, and it gets inside of these enzymes and plugs them up. And when it plugs them up, it makes it so we can't do anything anymore, and enzymes stop working. Great.